Yeah, I'm Mauricio Terrones. I'm professor of physics, chemistry, and material science and engineering. Here we basically grow carbon nanotubes, and we use aligned carbon nanotubes as a filter in order to trap viruses of the same size. Flu viruses have diameters of 100, 110 nanometers. Zika virus has around 65 nanometers. So the idea is that we can trap viruses by size. So by flushing with a syringe, the virus, we collect the virus, you can then flush it, and we can collect them in this very small tiny space, but we can collect lots of viruses. And then once we collect them, we can identify them. And actually we don't need an antibody, we don't need any label. And that has been the success of this research and this work. And we are, based on this research and work, we are just starting a new company, which is called Virolog Technologies LLC. And the idea is trying to make and commercialize these sort of devices or cartridges to identify viruses of different types, which could be related to plants, could be animals, or could be human viruses, but we can have an early detection without any antibody. And this is one of the things that we're doing right now in the College of Science.
Yeah, my name is Benjamin Lair, and I'm an associate professor of chemistry at Penn State. The project that I taught with the board dealt with working on new means of polymerizing polymers. In particular, when you think about polymerizing polymers, there's two broad classes, one of which is polymerized by shining light and generating electrons. That's a technology that's pretty mature for 3D printing and on-demand printing, on-demand curing of polymers. And so the project we're working on is trying to figure out how to bring that to a new class of polymers that is cured thermally instead of typically by shining light on them. As far as applications go, I think that there's several broad areas. One is medical devices, and so these are the types of polymers that people sometimes use for implantable devices like stents. So the ability to rapidly generate devices that would be implantable has utility there. But there's also other areas. These are polymers that are used in regular household goods, and so you could imagine printing replacement parts for things that break around the house, or I guess printing toys for children on demand, which could be good. They're also used in paints. We've cured polymers that are used on clear coats on cars, and so that's another area where there could be a lot of advantages.
So I'm Yu Zhang from Department of Statistics at Penn State, and my research is in methodology development for biochemical problems and large data analysis. And so the technology I'm developing is a computer algorithm to integrate functional genomic data sets to predict disease mutations and understand their functions in a disease. And so what we did, first of all, is to gather functional data sets from the public domains. There are thousands of experimental data sets available right now that all the people can use, but then the question is how to best use them. And so the algorithm I developed is to identify active regulatory functions in a human cell type. And there are hundreds of human cell types. So we identify functions within each of those cell types and try to connect the functions that we predicted to the disease mutations we identified from our genetic studies. And so the question is, for the mutations we identified, what are their potential functions? And in which specific human cell type they might have impact. And that's exactly what this technology is trying to address. And this is a challenging problem, but this will make a huge contribution to the potential target development for the drugs and leads to a better treatment for disease.
My name is OBrien. I'm an assistant professor in the Department of Chemistry. Our technology is based on some new fundamental discoveries concerning the role of kinetics in protein synthesis. We have a physical science lab that develops theories and computational techniques that took advantage of these discoveries to rationally redesign mRNA sequences in a new and novel way to control different aspects of protein behavior and synthesis. The benefit of this technology in the long run, we believe, will be to lead to greater and more efficient biotechnology processes to enhance different aspects of protein efficacy.
My name is Gong Chan. I'm a professor of biology department and Brian M. William Chair in life sciences. So what we invented is a new brain repair technology. Unlike many other published work or more people familiar with, where people put stem cells into the brain or into the spinal cord, we invented a technology using the brain internal glial cells that surrounded the neurons to change them into neurons for brain repair. One recent work we just finished is a stroke. So take a stroke, for example, when the blood vessel got clogged inside of the brain, and then brain will have this ischemic damage, lack of oxygen, lack of nutrient. Many neurons will die. Of course, a lot of glial cells will die, but glial cells are different from neurons. Neurons cannot divide, but glial cells can. So after neurons die, after some glial cells die, the remaining surviving glial cells can divide and divide, form this glial scar. And that is initially was preventing the injury from spreading. But eventually this glial scar long-term stay would prevent the other neuron to get into this injury site. So that's why it can cause the permanent disability of the patient. We invented technology, we inject a neurotranscription factor called NeuroD1 into this glial scar. We can essentially change the glial cell directly into neuron inside the brain, inside the injury areas, without any external cells, without any stem cells. So this technology, I call it a next generation for regenerative medicine, because we can now use patient internal cells, regenerate new neurons. And recently we quantified the efficiency of our technology. We can generate 100 times more than the internal capability. Most of other people publish can generate a few neurons per square millimeter. We can generate several hundred new neurons per square millimeter. So that's the difference. I believe our technology can be used not only to treat stroke, but also Alzheimer's disease, Parkinson's disease, Huntington disease, spinal cord injury, ALS. The list can go on and on.